Hello, Amy.
Hi, Kyle. How are you doing?
Good. How are you?
Good, thank you. Excellent presentation.
Thank you.
Oh, hi, Lay as well.
Wow, a lot of people are joining. Okay, cool.
Yeah. Lots of interest in your work. We'll just maybe give it a couple of
Yeah,
minutes.
let's get like a minute or two for
Yeah. All right. Well, looks like we have a good group to start anyway here, and people are still trickling in. Maybe we'll just wait for that countdown, the 28 unassigned participants, to get a little bit lower.
Sure.
People are still moving into their rooms. Okay, well, why don't we get started? And yeah, so hi everyone. Thank you so much for joining the Proteus competition and learning more about the launch event, and also coming to this breakout room on the antibacterial phage hydrogels. I'm Amy Hector. I work at the McMaster Industry Liaison Office, and I'm very pleased to introduce Kyle Jackson and Lei Tian to you. They're two of the inventors on the technology that is part of the Proteus competition this year, and they're here to answer any questions that you might have related to the technology. So why don't we start by just seeing if there's any questions in the room. Feel free to take yourself off mute, or you can also write questions in the chat. Either works for me, and I can help to facilitate some of the questions.
Okay. I actually have a general question. Did anyone actually know what a bacteriophage was before I introduced it today?
No, not me.
Yeah.
Okay, so there's a few people. Okay, I'm actually kind of happy.
Yeah.
When I speak at some of these kind of innovation things, a lot of people are looking at me like, "What is a bacteriophage? I have never heard of that before." Okay, cool. Okay, sorry to interrupt Amy again.
No. It's a good start.
That's interesting. Do you think that for a person who doesn't understand what a bacteriophage is, do you think that would freak them out a little bit?
I think when they hear the word virus, yes.
Exactly. Yeah.
This has actually been kind of a long-standing issue in the phage research community, because yes, bacteriophages are viruses, but and you think right now because of COVID-19, there's more a stigma behind bacteriophages. It's actually the opposite. When I speak to government officials about bacteriophages in general, they actually know the distinction now between mammalian viruses like coronavirus or influenza and bacteria, and that there are prokaryotic viruses in the form of bacteriophages. So it's going a lot better. 10 years ago, like it was bad. Like people thought bacteriophages are bad viruses, but now it's actually fine.
I had a question about, just maybe to get the conversation going, what led you in this to develop something like this? What drives you, basically?
I will let Lei answer that because I joined the project after he did.
Yeah. So could you say again? The signal is not very good. So I could.
What drives you? Like why did you want to do it?
I think first, of course, is our supervisor, Dr. Semi Dost. Like her specialty is on bacteriophage. So we are trying to build a biomaterial based on that. So like try if we can combine biology with material aspects. So we like just compact with this hydrogel, we can compact a lot of like very high concentration of phage together to make more like a pill, like to treat like especially drug-resistant bacterial infections. That's the starting idea we got. And then like the first investigator is our master student, Asda. Like she successfully like concentrate phage a lot. Then we finally found, boom, it can actually solidify to like a solid like material from nanoparticles to actual like a hydrogel you can actually see and put it on your hand. Yeah, it's a very interesting process.
Yes.
Oh, hello.
Hello.
Hi, this is Susan from UO Grove. So I have a question. So what you invested is like a technology to make a hydrogel, like not to design the bacteria, like what's the name? Bacteria.
You can call it phage for sure.
Okay, yeah, phages. So basically we can put like whatever phages we want into that hydrogel, right? Or
Yes, that's a very good question. Like right now, the main material of that hydrogel is M13 phage. But like, of course, we can add other phages to it. We already have some unpublished data of that proving like after embedding different bacteria, we can kill in different, after adding different phage, we can killing like a lot of different bacteria, including like U157, which is a very common like food pathogen. Like pathogen, like you have like always like you can see reports about like 157 on lettuce, meat. That's the experiment we did, and also other drug resistant, like remember BL21, yeah.
oh, thank you.
So essentially, we're able to design personalized treatments for very specific situations. So as Lei said, that we can modify these biomaterials to attack E. coli 0157, which is what he says is a very common strain that results in many outbreaks on lettuce and other food. But we can also design these materials to target more clinically relevant strains such as methicillin-resistant Staphylococcus aureus or MRSA, which is a very common infection found in both prosthetic joint implants, or even when it comes to Pseudomonas aeruginosa infections like cystic fibrosis. So there are a lot of applications here, a lot of ability to amend and essentially design the gel for varying applications. So it's quite limitless, which is quite exciting for this technology.
Okay.
Hi, I had a question pertaining to kind of the bacteriophage's resistance to like the bacteria becoming resistant to it per se. One of the claims that you have is that like it can evolve with the target bacteria. So are you viewing that as like within the system, the bacteriophage is going to continue to be able to kill the bacteria regardless of the changes it has? Or are you looking to more so if a bacteria does develop resistance to the bacteriophage, you can go in and tinker with the bacteriophage you're using and then reapply that to that situation?
Do you want me to explain that?
You can do that. Yeah.
Okay, sure. To answer your question, yes to both. We can do both things. So the bacteriophages themselves, as I said, they are viruses, so they have a genetic makeup to them. So they will naturally evolve with their target bacterial host in order to survive. So remember in my presentation, I said that these viruses are very selective. That's both a good thing and a bad thing. So the good thing is that we're able to target different bacteria that we want to. The bad thing is that those are the only targets the virus can survive in. So in order for the virus to replicate and survive, which is its primary objective, it needs to evolve with the bacteria naturally in order to actually survive and keep propagating and stay alive essentially. But to your second point, we can go back into the technology itself and modify the material to incorporate new bacteriophages that we know will kill that target bacteria that has developed some sort of resistance to the original bacteriophage, because it does happen. Regardless of what what pharmaceutical intervention you do, whether that be in cancer therapy or that is in this space in antimicrobial therapy, there will be eventual some resistance to your existing therapeutic strategy. But the best thing to do is to proactively design mitigation strategies in order to minimize that risk. And for us, we have two strategies, that being the fact that these viruses will naturally evolve, and two, it's so easy to go back and tinker with the design of technology to overcome those resistant bacteria. Does that answer your question?
Yes, thank you very much.
Perfect.
It looks like,
question.
oh, sorry, go ahead.
Is this gel designed only for bacteriophage, or can it be used as sort of like a platform technology for other pharmacological agents or drugs or other applications outside just the M13 phage that you use?
That's a very good question. Like right now, like, of course, possible application is we still use phage as a building material. That's the basic foundation novelty of this project. If we switch it to other proteins, that could be another completely different topic first. And second, yeah, it's possible to load, for example, antibiotics or antibodies in the hydrogel to realize slow release or rapid release based on your demand. And so you can realize dual therapy. That's also a potential application for this.
Thank you. So the gel is optimized for bacteriophage as of now, right? If I'm reading it correctly?
Yes. Yeah.
Okay. Thank you.
Looks like we have a couple of questions in the chat. The first one is there any method to administer the gel non-topically?
Right now, the predominant applications that you foresee for the gel are on a topical level, burn wounds, for example, or if we wanted to coat a material with it, for example, an implant, like a joint or a hip implant, for example. We are actively looking at other potential applications of this technology, which we can talk about at a later point in this competition, if you want to go down those lines. Options for maybe aerosolized or even intravenous applications, but that can be a conversation for another time.
Excellent. And Andres, can I ask you to keep your hand up for 1 minute, and I'll just ask the other question that's in the chat for now. So the question is, on the business side of things in the future, you will be dealing with governments or reaching out to private sectors to sell the product. So I think the question is, will you be doing these sorts of things? And then the second question is related. It's, what problems are you facing on the business side of your project in general?
Okay, so to answer that first question, I'll back up a little and say the commercialization pathway for bacteriophages in Canada is very complex right now. I was actually on a meeting earlier today with a few bacteriophage companies talking about this exact issue. Bacteriophages, as it stands right now, for therapeutic context, are not approved for therapeutic treatment as like over the counter, for example. It's approved for compassionate use. Ergo, if you're in a dire situation, it's a last resort case, and then we can use bacteriophages from a therapeutic standpoint. But in terms of active use on a daily basis, it's still kind of wishy-washy. Now, in terms of commercialization opportunities from a government versus a private sector perspective, right now, the technology is partly owned by McMaster University, and we have the ability to license out to other companies for them to use our technology for whatever purposes they want to do. We actively have no interest really in developing a company at this very moment focusing on this technology commercialization, but that can easily change in the coming year or two, depending on where our next publications land and what the next future technology is like. To answer your second point, your second question about what problems we're facing, realistically, the business problems that anyone would face from the bacteriophage commercialization standpoint is any kind of problem that any other startup company would face, and that is trying to get regulatory approval to use this novel technology in the commercial market, trying to get funding, whether that be through angel investment, venture capital, or through some sort of research grant to actually finance the research itself, as well as just trying to hire more researchers and more actual development individuals to actually push the technology to commercial market. So it's just like any other startup company, really, in terms of the early problems that we have in pushing this technology to market. I know that's kind of a non-answer for you, but that's the best I got at this point.
And Kyle, maybe to follow up on that, related to the development of the technology itself, would there be any challenges that the technology itself might run into in order to scale up, for example, or anything related to that?
Yeah, two things. One, regulatory approval, because we still, in terms of bacteriophages alone, it's kind of wishy-washy as if they're classified as a drug, or if they're classified as a biologic, or if they're classified as something else entirely. It gets more complicated because now these bacteriophages are encapsulated in an actual biomaterial structure itself. So does that change the regulatory pathway from Health Canada or the FDA's perspective? In terms of other challenges, manufacturing is another one. So currently, as Lei mentioned a bit earlier, right now we need to kind of do a lot of hands-on work in terms of propagating bacteriophages up to very high concentration. So that includes making very, very large volumes of solution in a very tenuous and strenuous way. So developing some sort of automated manufacturing through a bioreactor and purification system to streamline that process to make it less variable and more consistent, as well as making sure that a purified bacteriophage solution to work with every single time. That would be very, very enticing. And if we could partner with an existing company, for example, to outsource that manufacturing, that would definitely streamline the scale up of this technology for sure.
Excellent, thank you. And Andreas, you've been very patient, thank you.
Oh, no worries, thanks. Can you hear me? Yeah.
Yes. Yeah.
Okay. I was just wondering, I don't know if you mentioned it before, but can it be used as a coating? And what would be like the compatibility with commonly used surfaces, like in the medical industry?
Lei, do you want to take this one?
You go ahead. I feel like you have more ideas on the industry, like the application for this part. Yeah.
Fair enough. So yes, it can be used as coating. We can make these hydrogels very thin, to be honest, and we have that capability, so we can coat the surface quite easily with it. We are actively exploring that in the lab, and because of that, we can coat like stainless steel, for example, that's very common in hip implants, for example, and that can easily fit into the surgical procedure and have that natural antimicrobial surface to prevent infection at the site of surgery, which will enable more speedy recovery of the patient itself and reduce risk of contamination and infection thereafter. Does that answer your question, or do you have a second component to your question, Andre?
Yeah, that's perfect.
Okay, perfect.
Thank you.
Yeah, of course, yeah.
Okay, don't be shy. If you have questions, feel free to put up your hand or come off mute.
Just a follow up, maybe.
For sure.
Is there a possibility to know what polymer are you using? What matrix?
Lei?
Okay, sure. Yeah, you're asking what polymer we use, like what's the metric? Yeah, like this.
Yeah, for the hydrogel. Yeah.
Yeah, like the entire hydrogel is made of phage. So there's no other polymers we add. Yeah. So like the phage is not only the antimicrobial agent, but also the building material. In the paper, like I shared in the chat, you guys see the interesting hierarchical structure of the hydrogel, which is brought by the phage. Yeah. Unfortunately, like we have, well, there's a very recent paper we just completed. We also checked their much more organized nanofibrous texture of the hydrogel. We probably will submit that by this month. So hopefully it will be published in January. At that time, like I will definitely share again of that paper. And for
Okay.
phage hydrogel, I'm going to post Kyle's review paper on phage hydrogel, so like you guys can get more like direct information about this, like including our work and other people's work. Yeah.
Perfect. Thank you.
And it looks like we have another good question in the chat. The question is, if you are using these kinds of gels and implants, how will you account for a potential immune response against the phages?
This is a very good question. So bacteriophages, in a nutshell, are just a collection of proteins, just like any other virus. And there will be some level of immune response against it. This is still kind of in terms of the literature and research out there, it's still a very wishy-washy area. Bacteriophages are harmless to mammalian cells. They will not kill those cells. But when administered into a patient, you will see elevations in antibody levels, primarily IgA and IgG antibody levels. But those increases in antibody proliferation actually do not elicit a strong enough reaction to actually demonstrate symptoms. So, because you have bacteriophages in your body right now, like there are trillions of these viruses circulating in your bloodstream and in your body right as we speak right now, and your body's not reacting to them. In fact, you probably actually have M13 bacteriophage inside your body right now as well, which is the key component of our technology. So the fact that you're not experiencing a deleterious immune response to the phage right now demonstrates that it is novel in terms of its cytocompatibility and biocompatibility and its ability to not elicit a strong immune response.
Excellent. That's really interesting. I actually had a similar question when you mentioned that the bacteriophage can actually replicate. So I was thinking, okay, if you're administering them to a patient of some sort and they continue to replicate, does that cause any negative effects on the patient?
We don't know yet. This is another thing that needs to be studied. And so like my main PhD project is looking at this specifically from an oral phage therapy standpoint, looking at trying to treat bacterial infections in the gut. And this is one of the components that I look at specifically. How does different dosing of bacteriophage therapeutics impact both therapeutic efficacy and potential accompanying immune response to that therapeutic solution? Because if you have remdesivir, for example, remdesivir is, I'm not sure if people follow a lot of COVID-19 news. Remdesivir is a antibody therapeutic cocktail that is designed to target SARS-CoV-2. Antibodies are biologics, they are proteins, so you will actually have a triggered immune response against those biological therapeutics as well. But it's still, in terms of like the cost benefit analysis, the pros and cons, the pros outweigh the cons because you're targeting the more dangerous pathogen that's triggering a more powerful immune response, as opposed to your own body's ability to try to attack that foreign protein in the form of the antibody. It's the exact same concept with bacteriophages. The bacteriophages are trying to target the primary causal agent of the immune reaction, in this case, the infection, even though the body will try to say, oh, hey, that's a foreign antigen. I don't know what that is. I want to kill it because that's what the body wants to do. But right now, the body is so preoccupied in trying to fight whatever bacterial infection that you're trying to kill, it's not going to care about the bacteriophage, to be quite honest. Or in turn, it might care, it might respond, but it's not going to care as much as the established infection.
Interesting. That's an interesting point. So I think we've had a few people come in and out of the room. So I'll just reiterate that for anybody who's new, if you have any questions at all, please feel free to come off mute, raise your hand, or type it in the chat, and we'd be happy to answer any questions that you have. And we have Kyle and Lei here, who are two inventors on the technology. I actually had another question for you both related to competitors. Do you know of any competitors, whether direct or indirect, that would be kind of competing with the work that you're doing here and potentially commercialization?
Yeah, I think Kyle has like direct information of that. You can give them some hint. Yeah.
Yeah, so there are two main research labs that are also doing this type of work in terms of commercial applications. There's a team down at MIT. I'm lapsing on the actual name, and then the other team is from, I'm trying to remember, University of Pavia. So both University of Pavia and MIT are both actively investigating bacteriophage hydrogels for various applications. Whether or not it's in the same space as what we're imagining for our technology is still to be determined, but they would be considered direct competitors. In terms of indirect competitors, it's essentially the entire pharmaceutical landscape, because you have different strategies to try to treat these antimicrobial resistant pathogens, whether that be from established companies such as Pfizer or Roche to more up and coming startup companies. I mean, there's been a few startup companies in the Hamilton region coming out of McMaster University looking at trying to design novel antibiotics and trying to modify antibiotics to minimize resistant profiles. So there's a lot of people trying to get into this space, but from very different approaches, which is really, really exciting to watch and to see all these unique perspectives. Because at the end of the day, we're trying to solve a common issue. And if we thought COVID-19 was bad, to be blunt, I don't want to scare you all. If we thought COVID-19 was bad, just wait until AMR gets worse. It's going to be quite terrifying, to be honest. And we're already seeing the issues pop up right now.
Okay, we have another question in the chat. It is, are you planning on creating phages that have wide tropism for different bacteria or ones that are specific?
Yeah, I just typed the answer in the chat. Yes. Like
got it.
we have made hybrids like hygel, like all microgels, and prove like their antimicrobial range of the target can be programmed by just selecting what phage we add. Yeah, that can be realized easily, which is obvious. Like we already got like a lot of information. We know what phages can kill what bacteria. So it's really just depends on what we select.
We also, to put on top of that, we also have the capability to use synthetic biology techniques to actually modify the genomes of these bacteriophage to target very, very specific bacteria. So there's two things we can do. We can just source natural bacteriophages from the environment, which is a conventional way of doing, or we can actually use molecular genetic techniques to modify these viruses to target very, very specific bacteria. Or in the case of an earlier example, we can actually go back and change the genetic structure to make sure that it overcomes resistance to that original bacterial target. So there's a lot of different techniques we can use to both overcome potential resistant patterns, but also to target novel emergent bacterial strains in the environment.
Excellent.
I have a question. Have you ever do a long-term experiment to see if the pathogen can get antimicrobial resistant to your phages hydrogel? Like, for example, we know the bacteria have their own immune system. For example, the CRISPR, they actually can uptake a piece of the DNA from the virus, and then when they next time meet the virus, they can recognize the virus and then get resistant. Like, have you ever done like similar experiments?
Yeah, that's a very good question. And yeah, I can see you have, you actually know a lot about microbiology to ask that. Yeah. So yes, like what that's a problem with phage as well, and the hydrogel itself didn't solve that question that much, but like well, we know like phage can can be mutant and evolve, but so would phage do like mutant and evolve as well based on the bacterial target. We did observe like resistant bacterial growth. Yeah, it is a one problem we're facing. Kyle, do you have more experience on this?
Yeah, I haven't done anything specifically to date. It is a very interesting route to go down. I will say that I'm glad you brought up CRISPR-Cas9 or just CRISPR-Cas system in general. It is highly variable between bacterial species. So just because one bacteria and its offspring and progeny will have some sort of resistance doesn't mean that the virus can then evolve to actually combat that, which is a true value from using these bacteriophages, because they will evolve to evade that CRISPR-Cas system. It might just take a bit of time, but eventually will become infective again. Mind you, as I said earlier, we do have the capacity to quickly modify the gels themselves, as well as the bacteriophages within the gels, to overcome those resistant mutants. So although yes, this is a concern that we do have mitigation strategies to overcome these risks.
Cool, thank you.
Okay. Don't be shy if you have any questions. Otherwise, I'll just keep asking as many as I can to try and get as much information as possible from Kyle and Lei tonight. Oh, did I hear? Oh, maybe not. Okay. I guess one question that I had was, how long do the antibacterial properties actually last with the phage gel? And do you need to store the phage at a certain temperature? Is there any restrictions related to that?
That's a very good question. Like, so right now we found the phage hydrogel is quite stable, which is the advantage of making the hydrogel rather than like keeping the normal phage. I used to have like samples I storage at room temperature, like in a sealed environment, like environment on my lab bench. And after a year, I can still feel I test it again, and it can still kill bacteria. Like yeah, which was very impressive and unexpected. But yeah, that's how. So yeah, at least can stay 1 year. Of course, like the standard method, I would recommend to storage at 4 degrees.
Oh, that's definitely a long time they're stable for, compared to other kind of products that would be required to be stored.
After a year, we test both the mechanical and the biological property and the hydrogel, and it's still stable.
Wow. Okay, Zara?
Hi, yes, I just had a quick question, and apologies if this is a bit repetitive, but I was just in another room. And so I do remember that you mentioned that this gel can only be used topically at the moment. And you also said that there are a lot of issues that we might need to overcome for like regulatory approval. So I was just wondering, any plans of making the hydrogel injectable, or is it just not something that's feasible?
I think it's possible. Kyle, do you have any comment?
Yeah, no, we are actively exploring those opportunities right now. It's a bit of a different kind of use of the technology, so we've had to change up the actual protocols in terms of how we design the gels themselves in order to meet those specific requirements of either IV injection or air spice, for example. But we are actively exploring that, and we can touch base if you're interested in working on this project. We can touch base with another time to talk about those applications in more depth. And then your second question is about regulatory stuff?
Yeah, so if you were to like make it injectable, would it make the regulatory process more complicated? I'm assuming it would.
Oh, yes.
Okay.
Very much so. I'm not sure how familiar you are with clinical trials.
Not very familiar, but
Yeah, so clinical trials for topical applications is much more streamlined and efficient compared to if you want to inject something into the body. It also costs an exponentially amount more money to do injectable type clinical trials. So it is very much an application that we want to pursue in the future. The question though is, do we have the resources internally to facilitate those type of studies? But more importantly, do we actually have the finances in order to actually pay for those studies? Those are both outstanding concerns that we have and that we need to solve before we can actually consider going down that road, unfortunately.
all right. Okay, thank you so much.
One thing that I do want to add onto something that Leigh said earlier about stability is that it's been interesting to see how stable the viruses are at different temperatures too. We have found that I mean, for example, like the image that I showed in the presentation, that gel was stored at a higher temperature compared to room temperature in order for it to actually show that bacterial killing. So the fact that the gel is able to kill still at elevated temperatures around 40-ish degrees Celsius really expands the applications because, for example, in Africa, you do have those larger temperatures, those higher temperatures you need to deal with. So in terms of storage applications for this therapeutic, we have demonstrated that we're able to store at those naturally high temperatures seen in Northern Africa or the Middle East or in some parts of Asia, that most conventional interventions, whether that be through antibiotics or even the vaccine supply right now, that was a huge issue earlier in 2021 about vaccine distribution to these harder regions of the globe. That's really not a huge issue for us because these bacteriophages are stored so well within the gel. So that's a really novel application of our technology as well.
That's a very interesting direction. Like maybe a solution is harvesting phage from Africa. That would be definitely temperature like resistant. Yeah.
They do exist. Phage that are resistant temperature do exist in Africa. And I do know that the phage community in Africa is booming right now. So that would definitely be an option to go down.
Excellent. Okay, we have a new question in the chat. What kind of environment can you use these gels? Does it have to be extremely sterile environment like hospitals, or can you use it in frontline, example, in rural areas of third world countries?
I mean, when you storage these samples, I would definitely recommend to seal it in a sterile environment, like the container. But when you use it, I don't think there is like strict rule of the environment you need to use. Yeah.
Yeah, for sure. And another kind of pathway that we have thought about too is defense route, partnering with the Canadian Armed Forces or the U.S. military, for example, to apply this as a battlefield wound dressing type application because it's a hydrogel. You know, it can actually absorb water, or in this case, blood loss from a wound, but it's also naturally antimicrobial, so we can actually prevent further infection from that wound occurring. So we can use these types of technologies on the front lines and also in the hospital to answer your question specifically.
Yeah, that was a really good question, and actually that was one of the things that I was trying to think about with your technology is related to the applications. What some of those different uses of both the structural aspect and the self-healing aspect could be used for, because it seems like there's kind of almost limitless.
Yeah, no, the self-healing aspect is really crucial because I'm sure everyone here has probably had a Band-Aid on them at some point, and like the mobility, for example, with a Band-Aid, say you have like a cut on your arm here, if you move it around, that Band-Aid might actually impede your ability to rotate your arm. But because the gel is a soft bond material, because it's flexible and can actually self-heal, if you move your arm and it tears, with a Band-Aid, you gotta replace it. But with our technology, because it tears and because it's self-healing, when you put your arm back into a natural position, it will readily self-heal and continue blocking whatever potential blood loss you might have if you have a wound, for example, which it'll absorb that fluid. So there's a lot of intrinsic value to these gels that are more valuable compared to conventional band-aids or other things currently in the market.
Very cool.
Yeah.
Okay, another question in the chat. In the background provided about the project, you mentioned imaging application with fluorescence. Can you expand on this a bit?
Yeah, like so far we found the hydrogel itself is autofluorescence. This signal is not generated by phage itself or the crosslinker. It's really the crosslinking reaction, like the chemical reaction brought the phage to be connected. That's the reaction caused for example, it forms like carbon-nitrogen double bond and other similar like chemical bonds that can generate fluorescence. We are not very sure about the fluorescent application yet. That's just a potential direction. I would not recommend it to explore that much about this because right now we don't have any clear like idea about this.
Del, you're asking some excellent questions tonight. So well done. Any other questions from the group here? No. Okay. So you've mentioned a couple of times that the technology is literally phage in water. Is there anything that would cause toxicity or anything like that to a human in the compound that you've developed?
That's a very good question. Like right now, like one concern we all have is the crosslinker we use, which is glutathione. It's cytotoxicity, but like it precipitates the crosslinking reaction. So like most of them are consumed, and because it's in water, like we washed it at least 3 times to minimize it, and we found at least the phage itself. We haven't done the cytotoxicity test yet, but at least based on the published papers on the similar evaluation, the final product is pretty human friendly, and we haven't observed that hydrogel itself has toxicity to other like bacteria that M13 can't kill, which kind of proves that here, like the glutathione itself, the final product is safe. And another possible way to avoid this concern is using different crosslinker, which we already done work. There are other crosslinkers we can use to construct this hydrogel, like connect phages together to form hydrogel.
So thank you. And Taha, you had a question?
Yeah, so I want to try my best to phrase this question, but it's kind of like I cannot figure out how to do it in my head. But basically, I remember you guys mentioned like the stat at the very beginning about the World Health Organization saying that there's going to be about 10 million people who are going to die due to like AMR related infections. And you guys also mentioned like the market cap for essentially antibacterial compounds growing from like 38 billion to like 55.8 billion in like 2023. Now, what I'm trying to, I guess, ask is how to, I guess, bridge these two different estimates. And what I essentially mean by that is, can you guys, I guess, estimate economically the demand that would be created based on those rise in AMR infections as they pertain to the increase in antibacterial phage when it comes to their demand? Because I'm not very like experienced when it comes to like biotech or just in general the selling of antibacterial hydrogels. So I guess in general, where do you guys see, I guess, demand going for a product like this? Because it is the first of its kind.
That is a great question. I think the best way to answer that question is with another statistic. Last year in 2020, the United States spent $250 billion on antibacterial treatment of their citizens. So that was in the United States alone in terms of their cost to their healthcare. I think their healthcare spending every year is like $2 trillion. So about an eighth of their healthcare was spent on trying to combat AMR infections, which is a staggering number. So by year 2050, when this is estimated to be an absolute global catastrophe, I'm trying not to be pessimistic here or anything like that, but the actual market demand is going to just explode. I don't think there's any way to quantifiably estimate that market demand because we're already seeing the effects right now. And as I mentioned earlier during my presentation, COVID-19 has exacerbated those effects. So the demand for these novel solutions will be even greater much sooner. I can probably foresee that the market cap will reach 100 billion.
Hi, everybody. My name is Tina Centris. I'm from the University of Windsor and I'm on the organizing committee. I don't see Suzanne in the meeting right now, so I guess I'll just take over for hosting. But we want to thank everybody for joining our virtual launch opening for the Proteus Innovation Competition. If you guys do have any questions, more information can be found on the Proteus website, as well as there should be a form on there and some of our contact information if you have questions about the competition or any of the technologies that were presented today. I want to remind everybody that abstracts are due, I believe it's January 10th. As well, you can register your team on the competition website, which I can put in the chat. And then the respective dates for the rest of the competition are posted on the website. So thank you so much for joining. I guess myself and Amy, who's on the organizing committee, can stick around, but thank you for joining.
Tina, I have a question before I leave. I was wondering if there will be another event that we can partner with someone else who's also interested in the technology.
If you want to form a team, but you don't have one for yourself, what you can do is on the registration page, you can click that you don't have a team that you'd like to be matched. Everyone else who clicked that setting will be then put into, I guess, an email thread, and then will be left to kind of form their own groups that way. So if you want, you can do that. Or you can reach out to Jordan, who is the conference marketing coordinator, and he can help you find a group. Oh, there's Jordan.
Yes. So yeah, if you want, you can fill out the form, or you can just send an email to proteusic@uwo.ca, and we'll do our best to pair you up with a team.
All right, that sounds good. Thank you so much, Jordan and Tina.
So there's another question in the chat that says how many people have to be in a team. You can be a team of one if you would like. But in the past, what we found is the teams that are the most successful have between 2 and 4 people, or maybe 6, but usually 4. So pick your friends wisely, I guess, and try and make sure that your group is well-rounded in terms of skill sets. And usually those are the teams that come up with the best final plans. You're welcome.
I just wanted to ask a question.
Sure.
Just what was the email you said again? Like, if we're looking for team members that we could send an email to, and my
proteusic@uwo.ca. I'll just throw it in the chat.
Okay, thank you.
Can the technology be changed after registration? I don't think so. Jordan, can they submit multiple abstracts though?
No. So basically, if you have the opportunity to change your technology up until the business plan. So if you submit an abstract for one technology, but you decide after further investigation that that technology isn't for you, you would just simply switch the technology that you prepare the business plan for. After the business plan, then you're stuck with that technology.
So I suppose we can stay on the line for the next 5 minutes. The event is scheduled to end at 6. So if you do have any other competition related questions, we can answer them now. Most of the information is found on the competition website. So that will be available to you the whole duration of the competition. So you can reference that.
I'm looking at the website right now. I'm not sure if the requirements for the abstract is mentioned here, or I'm not looking.
If you look at the competition outline tab on the top, and then scroll to stage 1, it says abstract. Underneath that, it'll say submission requirements. And there's mostly 2 sections. There's a please describe how the technology works section, and then there's a who is your target market and what will they use it for. And each one of those sections have a limit of 250 words.
That's good, thank you. And if we had further questions to write up the abstract, are we to meet with the inventors directly, or should we contact World Discoveries or portals to get that information?
I think it depends on the nature of your question. If the nature of your question is about the competition criteria or how it works, I think the Proteus email that's in the chat would be better. If your question is around a specific technology or parameter around the technology, then I would say to reach out to the respective lead from that institution and ask them their question.
Yeah, and if you don't have that contact, you can reach out to Jordan and he'd be able to put you in touch with us.
There's another question in the chat that says, can we meet in person or just by mail and calls? I would say that depends again on what your question is and who you're trying to reach out to. A lot of the universities have gone back to some in-person work. So if the person who you're looking at is at your own respective institution, you could certainly try and meet in person. But otherwise, I would say probably email and if a Zoom call, if they're willing to have one with you. Jordan, I think this next one could be for you. Do all participants need to be students or can they be recent alumni?
So the competition is open to the public. So alumni, students, and even just entrepreneurs in the community are allowed to join. As long as you're above the age of 18, you're welcome to join the competition.
And I was wondering also if this is a yearly competition and we will be repeated next year as well.
Yes, so the competition has been running for 7 years and we will probably be running it again next year.
Yeah, thanks.
There was another question in the chat of if they can find a recording of this presentation. Jordan will probably be posting those online.
Yes, so we'll be sending out a recording of the presentation here probably next week.
The next question is, are there samples of abstracts or business plans from previous years available? I don't believe there are, but the winning pitches from the previous years, some of them have videos posted on the competition website under the top tab called past years. And so that'll give you kind of a level of indication of what the quality, I guess, or a breadth that each team kind of covered in their final winning pitch. Sorry, I was just going to say most people, as long as they meet the criteria for the abstracts, proceed forward. It's in the business plan stage and then getting to the final pitch that most people are kind of more selected for, I suppose.
Thanks. And is the final pitch online or in person?
It will be virtual. So we haven't decided whether it will be a live event or whether teams will record their presentations and submit them. We're still kind of sorting that out. There's another question in the chat that says, when's the last day for abstract submission? January 10th, also found on the competition website. I believe it's midnight, January 10th.
Yes.
So we have time for, I think, one more question, and then we'll probably cut it. Well, I don't hear anything or see anyone typing, so thank you so much for everyone joining us. Best of luck in the competition, and I guess we will be in touch soon for the next events as part of Proteus. So thank you.
Thanks, everyone.